Good morning, everyone. Welcome and thank you for attending the IMG webinar series. So first, let me introduce myself and my colleagues who will be conducting this webinar. My name is Natalia Ivanova, and I'm a scientist at the Prokaryotic Super Program of the DOE Joint Genome Institute. And with me today are other scientists and software developers at the JGI. They are Rekha Seshadri, Amy Chen, Ken Chu, Neha Vardiz, Reddy, Simon Rue, and Emily Illa-Fedros. So first, let me say a few words about the format of this webinar. This webinar will be recorded and we will post it on YouTube. You will get an email with the link to the recording, and also the same link will be posted on the webinar homepage, the same page that you used to register. Your mics will be muted for this webinar, but you are most welcome to ask questions via Zoom chat. So, and the answers will be also via the same Zoom chat, hopefully real time. If there are many participants asking essentially the same question, then it will be answered by my colleagues on air. We will edit this chat transcript, add answers if they were deficient in some way, and we will post it together with the recording on YouTube, so you will have access to it later. In addition, basically, I wanted to say that we are going to conduct a live demo, which means that although we timed it and rehearsed it, and we sort of know how long it is supposed to take, there are some variables that are out of our control, such as the network speed. So if it runs over time a little bit, we apologize. So one more announcement that I have to make is that unfortunately tonight and tomorrow we have a JGI-wide compute system maintenance. It means that IMG and other JGI systems will be unavailable starting 9 p.m. Pacific time today, April 21st, continuing into tomorrow. And we hope that all of the IMG, all of the JGI systems, including IMG, will be up and running on Thursday, April 23rd, 2020. Again, we apologize for the inconvenience. And now let me start with the slides. First, let me say a few words about who we are. We represent the DOE Joint Genome Institute. It is a science user facility funded by the US Department of Energy Office of Science. And our mission is to advance genomics and other omics technologies to promote research in the areas relevant to the DOE mission, which is clean energy generation, environmental cleanup, and other environmental challenges. So we generate data to support plant, microbial, including fungal and algal science, as well as the studies of microbial communities by the scientists worldwide. So, and here are a few relevant facts about the DOE. So the next slide is about IMG. What is IMG? IMG is a system, JGI system, which hosts the sequence data and annotations generated by the microbial and metagenome programs at the JGI. It also has a lot of actually analysis tools to assist with analysis of this data. In addition to the JGI generated data, IMG also hosts relevant data to support these analyses. And these are reference genomes and a subset of publicly generated metagenomes from DOE relevant environments. IMG exists in several different flavors, and these are shown here. And the first one is IMG-M, which hosts only public data, does not require login, and has a limited set of tools. The second one is the one which I will be using today for the demo. It is called IMG-MER. It requires login. It hosts both private and public data, and it has a much larger set of tools. The next two are the specific systems for analysis of biosynthetic gene clusters. This is IMG-ABC, and viruses. This is IMG-VR. And I would also like to point out that IMG does support annotation and analysis of non-GGI generated data. You can do this by using this submit dataset link. Again, as a reminder, I am going to use IMG MER today. And the reason why is because download capabilities are enabled only in IMG MER. You need an IMG account to do this. So if you have difficulty either getting an IMG account or logging into IMG using this account, please go to the Contact Us link and submit a ticket. These Contact Us links are found on the bottom of the IMG pages, all of them actually. Unfortunately, it is cut out of this screenshot, but it is in the left bottom corner. It is also found in this help section, which is in the right upper corner. If you click on this help menu, there will be a section Contact Us and Other. And that section will have this Contact Us link. So let's say that you have an IMG account and you believe that you are logged into IMG MER. How can you figure out that this is the case? If you are logged into IMG MER, you would have your name on top of the screen, as well as logout link next to it. Also, you will have this workspace option in the IMG main menu. Another note that I would like to make is that I will use an example from the previous webinar conducted by Rekha. You can find the record, if you didn't attend it, you can find the recording and the transcript chat here on the same website that you used to register. It says this is April 14th webinar. Here is the recording and Q&A, and you are most welcome to watch it. Now let's look at what kind of data IMG has. So as a very gross oversimplification, IMG has two types of data. And this is the sequence data and also the metadata about this sequence data. And sequence data is mostly stored in these FASTA files, which exist in two flavors: amino acid FASTA files of protein coding genes and nucleotide FASTA files of the metagenomes and genomes, the entire assemblies, individual contexts and scaffolds, as well as individual genes and intergenic regions. The metadata about this sequence data falls into several categories. The first one is this GOLD metadata. And this was extensively covered during the previous webinar by Rekha. Again, if you haven't seen it, I encourage you to go and watch. GOLD records metadata essentially about the methods that were used to generate the sequences that you see in IMG. It also has the data about environmental parameters and environmental classification of the sample that was sequenced. And also it includes information about the taxonomy of the organisms that were sequenced. The next category of metadata is all kinds of cumulative statistics, such as the total sequence length, the GC content, and the count of predicted features, and so on. The next two categories are functional annotations and structural annotations. And these are essentially, these are showing you which regions on the nucleotide sequence encode for certain proteins, RNAs, and so on. And functional annotations are mostly found in the form of assignment of protein coding genes to the different protein family classifications, such as PFAMs, COGS, TIGR-FAMs, and so on. And as you can see, we have two options in IMG, and one of them is export, and the second is download. And the distinction between the two is that for export, you can essentially configure the output. You can select which specific data you want to see in the output, and also you will get the results in real time. While for download, we usually provide prepackaged data in a download bundle, and usually you are notified about when this bundle is available and when you can get the link and you can follow it to download the data. The difference between the two is the size of the data that you are trying to get from IMG. Exporting is usually very small files. For instance, environmental metadata even for hundreds of metagenomes is very small file. While an entire assembly of a metagenome plus all of the protein coding sequences, plus all of their functional annotations, is a file of hundreds of gigabytes. So the next important slide is the IMG download and export file formats. The most common one is this tab delimited text, which applies to all kinds of environmental metadata, functional annotations, and so on. If you are exporting it from IMG, you can specify the columns and you will see the headers of the columns in this file. If it is a prepackaged bundle download, the type of the data in the file will be specified by the file name, and the column definitions will be provided in a README file. The second common format is FASTA format, which looks something like this. It is sequence storage format, which has the sequence itself and the definition line or the header, which is designated by this greater sign. So this is common format used by many people. The only comment about it is that usually protein FASTA files have in IMG have extension .faa, and nucleotide FASTA files would have extension .fna. So as you can see, there is no FASTQ format mentioned here. And the reason for that is that IMG does not does not use raw read files, where in addition to the sequence itself, you also have the quality of each and every base. So the third format, which is common for IMG, is this GFF, General Feature Format. Again, this is a community standard format. We didn't invent it. And the developers of this format is the Sequence Ontology Consortium. There are standard column definitions, specifications, and so on, and these are available on GitHub and some other sites such as Gmod. The link to the GitHub specification of GFF format is here, and the files in GFF format would have the extension of GFF. Two more important topics about IMG data usage policy. So even if you go to IMGM, where all of the data is public, it doesn't mean that there are no restrictions on their use. So IMG data usage policy follow the JGI data release and usage policy, and they can be found here. You are most welcome to go and read and review this document. As a brief summary, IMG has three types of data. Unreserved public data, and this comes from the downloads from NCBI. It also comes from the non-JGI generated data sets that were submitted by private submitters and later released by them to the general public. And also it includes these published JGI data sets. The second category is reserved public data. It means that it is publicly available, but it has certain use restrictions. And lastly, there is the private data that is not available kind of for the general scientific community. Now let's focus on this category of reserved public data, which is essentially only one item, unpublished released JGI data sets. What does it mean? It means that even though the data set has been made public, the PI, the person who submitted the proposal, who generated the samples and sent them to the JGI for sequencing, reserves the right of first publication. This means that you cannot publish a comprehensive analysis of genes or genomic data prior to this first publication by the PI without the consent of the PI of the dataset. It also means that you cannot redistribute, you can download the data from the JGI, but you cannot redistribute or repackage the data without the permissions from the JGI, who will also ask the PI's consent. So to somewhat simplify for you the navigation between the IMG datasets and their data usage patterns, here is a small decision chart. So the question is, do I have to ask PI's permission to use the data that I see in IMG? If you have an IMG dataset, the first question is whether it is public or not. If it is not public, it is a private dataset and you must contact the PI and ask their permission. Unless, of course, you are the PI on the dataset. If the dataset is public, then the next question is, was it sequenced by the JGI or not? And this information would be found in the field Sequencing Center. If this field says Sequencing Center DOE Joint Genome Institute, then it is a JGI dataset and additional restrictions may apply. If it is non-JGI dataset, then it is free for use. So if it is a JGI dataset, the next question is, was it published or not? So in this case, you will review the information in this field is published. And if it is not published, then you will have to contact the PI and ask their permission. If the field is published is yes, then this is a published JGI dataset, and it has no restrictions on use other than the appropriate citation. We unfortunately, the PIs rarely, if ever, tell the JGI that they have published certain data and therefore now it is unrestricted use. Actually, all of this data about publications, it is mined by the GOLD team. And they are mining it from the publications where they're trying to match the PI's name in the author list to the IMG taxonomy ID or some other identifiers that is mentioned in this paper. And not in all cases, this matching is successful. So we make our best effort, but we cannot guarantee that all of this data that we have about publication is accurate. And now the last slide is the outline of the live demo that will follow. First, we will cover the batch download of IMG genomes and metagenomes. And it is important to note here that I am only talking about IMG data. If you are interested in how to download raw reads, how to download assembly, mapping of the reads like a BAM file, that is a topic of a completely different discussion. So in addition to this batch download, we will also review how to get the data that will help you with the this decision chart that was shown here. How to get this metadata and how to export them in a tab-delimited format. The second topic would be a demonstration of how to get gene metadata from the gene card and also how to download protein and nucleotide sequences of a selected small set of genes, including their upstream, downstream regions and other information. So that's the end of the introduction. And if Rekha and Amy, if there are any questions that we need to address immediately, please do so.
Yes, there are a lot of questions asked about downloading, and my reply is then Natalia will explain more, so please be patient. Thank you.
There was also a question about asking about how do you figure out if something has been published? And I said that you would go over that in a second, but there is a piece of metadata that says is published in the UI that I think Natalia is going to review in a bit. But there's some caveats associated with using that particular field.
That is absolutely accurate. That is what we are going to do. Anyway.
Natalia Marino asked, what about using data from gene neighborhoods? Where does that fall?
That won't be covered today. So there are several tools in IMG that have that essentially let you analyze the concerned gene neighborhoods, but that is off topic today. If you want this answered, please submit, basically, please respond to the IMG, to the survey that we will send you and put it as a topic that you want presented. Okay, then we will proceed with the live demo. So in the live demo, as you can see, I hope that you can see my screen. I am going to use the example, one of the examples that Rekha presented during the previous webinar. So I will go to Find Genomes menu to the Genome Search. If I click on it, I will get a menu that allows me to search for, to look for the genomes and metagenomes by different parameters. And I hope that IMG behaves today and the network behaves today and everything is fast. But again, as I said, it's live demo, so be patient with me. So this is the genome search that was shown during the previous webinar. And on the bottom of this genome search, there is the search history. So now, in order to make this presentation completely live, what I will do is I will clear all my search history and start from scratch. So normally when you get to IMG, let me reload this page again. Normally when you get to IMG, when you log into IMG, and again, I know that I'm an IMG MAR because I have my name, my username on top, and this logout link. And I also have this workspace option in the main menu. Normally when you get into IMG and you are a new user, you have this panel and there is nothing in your search history. So I am going to go to the, but there are some examples that you can use to populate your search history. To do that, you will click on this public list link, where you can find the pre-saved queries, these example queries that you can use to populate your search history. Right now in this list, there are only 3 of them. It may grow. And the first 2 queries on this list are those ones that were discussed in detail in the document that was sent to you, this genome search tips and tricks. So the 3rd query is the one that Rekha used during the demo last time. I will select this query and I will click on Add Selected to History card. And this will populate my history search. My search history in the genome search. So this link redirects me back to the genome search. And now I have my search history populated. This is the query that I used last time. And this query is looking for isolate genomes from the genus Bradyrhizobium that have no more than 500 scaffolds or contigs, and that have environmental classification with any of these 5 terms: nodule, rhizo, plant, stem, and so on. So I have 2 options here. I can either click on search, in which case it will bring me the results, or I can reconstruct this query so it will show up in the query builder. If you scroll up, this is the query that that Rekha used last time. So let's evaluate this query. Clicking on evaluate query, it runs the query without sort of bringing the results. It tells you which particular fields are being searched and how many entries were found in these particular fields. And this helps you to refine the query, because sometimes you find too many data sets, sometimes you find 0. And this is exactly what you would use in order to refine your query to get an approximately the number of data sets that you expect. So it shows you very, very detailed list of the things that of the fields that it's searched and how many counts it found for each of them. And the total count of these data sets is 85. And if you are following me in this presentation, most likely your count is different, probably less. And the reason for that is because I have access to several private data sets that you can see. Now let's modify this query. And that's one of the reasons why we store these queries, save them in the history, and so on and so on. Because you can reconstruct the query, and after that, you can modify it. So I am going to modify it by restricting the search only to the public data sets. Again, I am well versed in the IMG UI, so I know that this information about whether the genome is public or not is found in this category, sequencing, assembly, and annotation. And the field name is called isPublic. And I will select isPublic yes, meaning that it is publicly available and you should access to it as well. If you are new to IMG and you are not familiar with IMG search fields and categories, you can find the list of all of them down here. So now that I modified the query, I will click on search. And I expect that now the list will include only public genomes. And hopefully this time our counts will be the same, my count and your count. Again, IMG shows you very detailed information about how exactly this query was executed, which fields were searched, how many matches it found in these fields, and so on. So as you can see, now I have only 69 genomes instead of the 85 in the previous query. This is because only these 69 are publicly available. Now let's select all of them by clicking on Select All and add them to Genome Card by clicking on Add Selected to Genome Card. So now I am in my genome card, and it tells me that I have 69 genomes in my card. Also, the contents of your analysis cards, including genomes, is found here on top of the IMG main menu. Now, remember the slides that I showed you? All of these genomes are public. So in principle, I can use them in my analysis and publication, except that for some of them, for those that are GGI genomes and they are unpublished, I would have to ask the permission of the PI to use the data. So how can I go through this list of 69 genomes and figure out which one of them are free for use without any restrictions and which ones of them are restricted? So if you remember, the very first thing that we looked at was the sequencing center. Are the data generated by the GGI or are they generated by some other sequencing center? We can find the genomes that were not generated by the GGI using a filtering options found here, filter column. And although Reka showed it last time, I'm going to show a somewhat more sophisticated filtering that may not be obvious to the users. So I want to filter the column which is called sequencing center, and I want to exclude any entries that have this DOE Joint Genome Institute in them. How would I do that? First, I will select the column sequencing center here. Next, I will change the filtering options from text to regular expression. And if you look at it, there is this question mark here. And this is actually a tooltip. If you click on it, it opens a little window which tells you that filter text is plain text search and a regular expression search has syntax, which is described here. This is Perl regular expression syntax. If you click on it, it will open this page with the description of Perl regular expression syntax. And here is some of it. So if you put a dot in there, it matches any single character. If you put a caret sign, it matches the beginning of the string, beginning of the line, and so on and so on. So these are common regular expressions used not only in Perl, but also in Unix command line tools, in Python, in awk, in sed, and so on. And many people are familiar with them. If you are not, I encourage you to get familiar because this is really powerful search and filtering tools. Now let's go back to IMG and put a regular expression that would select for, that will reject for us actually the non-GGI, the GGI genomes. And this regular expression will look like this. So it looks like a lot of nonsense, but actually what I am selecting is the rows in the table that have that do not have capital D as their first letter in the sequencing center column. So this is what the first caret sign and the second expression in the brackets is doing. Next, I have to click on apply. And as you can see, all of the entries for the DOE Joint Genome Institute disappeared, which is exactly what I wanted to do. I wanted non-GGI genomes that have no restrictions on use. So now let's say that I want to save them. In order to do that, I can click on Select All, which will select all of these remaining rows. And you can check how many of them are here. Because if you scroll to the far right, it tells you that it selected 26 rows. Now remember that I had a total of 69 genomes in my genome card. So 26 genomes of these are non-GGI genomes, and they are free for use without any restrictions. In order to save them on my laptop, I would click on this export button here. This will generate a tab delimited text file, which can be opened in Excel. It will have this standard extension XLS, which Windows recognizes as a file for Excel, to be opened with Excel. So I click on OK. And when Excel will try to open this file, it will actually complain, because the format of the file is not a native Excel format. Rather, it is tab delimited format file. It is not corrupted, it is not unsafe, it is absolutely fine. So you can click on yes. And you see the content of the table in my genome card in this Excel file. So all of the names, IDs, and so on, including this very important column, the first column, which is called taxon OID. These numbers here in this column, these are the unique identifier assigned by the IMG to each and every data set it has. And because it is a unique numeric identifier, you can go back to IMG, upload this file, and your genome card will be repopulated with these genomes.
Natalia, sorry to interrupt, but we have a bunch of people saying that they can't see the table. Looks like the Excel file is not displaying on the screen.
Yeah, I have to reshare. I'm sorry.
Or the screen got frozen.
Yeah.
Working on it, guys.
Yeah, sorry about that. That's the wonders of Zoom. So this is the Excel file. Can you see it now?
Yes, Natalia.
Okay, great. So this is the Excel file, which is essentially which has all of the information that was in your genome card. In the important column, this one is taxon OID. This is the unique numerical identifier assigned to every IMG data set, with which you can find it later on, and which you can load into genome card to see it populated with these genomes again. Okay. So unfortunately, now I will stop sharing this screen, and I will have to go back to sharing the IMG screen again. So bear with me. Can you see the IMG screen?
Yes, Natalia.
Okay, great. So now we have addressed the question. So these are non-GGI generated data, public, free for use, no problem at all. Now that I have saved them on my laptop, I can remove them from the genome card by clicking on this remove selected. So after I remove these genomes, non-GGI genomes, I assume that all of the genomes in my genome card, the remaining ones, would be the GGI generated data. As you can see now, I have only 43 genomes in my card because I removed the other 26. Now, how can I make sure that all of the genomes remaining in my card are generated indeed by the GGI? They have this sequencing center, DOE, Joint Genome Institute. Well, you can sort this column back and forth. First, if you click first time on it, it will be sorted in ascending order, again, alphabetically in this case. So if I click on it, all I see in this column is DOE, Joint Genome Institute. If I click the second time on this header of the column, it will sort it this time in descending order, again alphabetically. And again, all of the entries that I see here are DOE Joint Genome Institute, so I am fairly certain that the only genomes in my genome card right now are generated by the GGI. And these are the data for which you have to make sure that they are published in order to use them freely without the permission of the PI. So how can I figure out which of these genomes are published and which of them are not? And also, if they're unpublished and I have to ask the PI's permission, how can I find this PI's contact information and name? So you can do this by using the table configuration options available on the bottom of this page. if we scroll down to the bottom of the page, you will find the table configuration options. So for the contact name and contact email, this is the PI's email and this is the PI's name. And I want them so that I know whom to write an email and ask for their permission. The second part of the information that I want is this, is it published or not? And I can scroll this table and I can try to look for it, and it is not very straightforward. In order to find it easier, what you can do is you can use the browser find option. I am in Firefox, and in Firefox, if I click on Ctrl F, this will open me this search box, little search box on the bottom of the page. And if I type here publish, it will lead me to the field in this configuration table is published. And this is exactly the field that I want, and I want to select it. So now that I selected all of the fields that I want to see in this table, I have to go up and I have to click on redisplay. Unless you click on this redisplay, IMG won't know that you want to add more columns to the table that you already have. So after you clicked on redisplay, let's scroll right and see that we have the columns that we wanted to see. So yes, now we have the column contact email. Here is one, for instance. We have contact name, which is the name of the PI. And we also have the column is published. And the first genome is unpublished, it turns out. Now, IMG, and it is a very long table, and you can get tired scrolling back and forth, left and right. So IMG offers you an additional option of how you can configure the view of this table for easier browsing. Here is this column selector option, which allows you to hide or show certain columns in this table. And in this case, let's say I don't care about domain or sequencing status or study name. I will still keep the sequencing center column. I don't care about genus or submission ID. I don't care about any of these identifiers. I will keep the is public column just to make sure, and I will definitely keep the columns for the PI's email and name. I don't care about environmental metadata. I do care about whether it is published or not. And I don't care about the rest of the columns. As you can see, as I'm clicking on this hide and show, the columns disappear. So now let's close this menu, the column selector menu, and see what we have. Now we have the table, which has only the data that can help us decide whether these genomes are unrestricted or whether you have to ask the PI's permission. So here is the column is published, and some of them have published no, and others have published yes. Let's look at one of them to see what kind of information is available in IMG that this published really means that there is a citation available for this genome. Let's click on the name of this genome and open it in a new tab. This is right click. So now I'm going to the genome details page of the genome with the name that I clicked on. Again, you can either use a browser find function, or you can just scroll down, and under this project geographic map, there is genome publication information. If you click on this cross, it will open this whole section. And as you can see, it is indeed published, and there is a citation with PubMed ID, with the title, and so on, and with the DOI that you can use in order to cite the data that you see in this genome. So that's how basically, this is why this genome is called is published yes, and somewhere in this paper there will be IMG taxon OID saying that this publication refers to this particular genome. Okay.
Natalia?
Yes.
May I interrupt? I'm afraid we have a couple or more questions asking
Okay.
about this usage of public data. So I have one person asking, they want to use an IMG data set to, I think it is a reference for assembly, and then I guess they want to publish. And then there's another person asking, is there statute of limitations on how long a PI can restrict the use of unpublished data?
And that's why we provided the link to the JGI data release and usage policy.
Right.
So again, it is a very long discussion, which is slightly off topic here. But the general kind of guidance is that if the data was generated for the proposal submitted before November 2018, then the PI has unrestricted, basically, for the lifetime of the genome, until it is published, or metagenome, until it is published for the lifetime, they have the right of first publication. That's how it is. For the proposals accepted after November 2018, they have 2 years for the first publication. After that, it is free for use. So for the using something as a reference assembly, the rule is still the same. If you are going to use it and publish it, you have to ask the PI. And again, so basically, please use IMG user survey if you find this information confusing, misleading, and so on and so on. This will help us guide and develop more webinars if we see that people are really confused and we need to clarify these topics. Is it okay, Reka, or do I have to say more?
No, I think that's fine. I re-shared the link to the data usage policy if you want to review it. Also gives you more details on, what type of analyses are reserved.
No, absolutely.
For PI. So it's,
okay.
It's not going to be like your one gene family or specific things. It's whole genome based analysis.
Okay. So now that basically, again, so let's go back to to this table. If I want to export it and save the metadata that I generated here, I will again select all of these genomes in my genome card and click on the same export button. This will again generate for me an Excel file, which will, this time will have the information about contact email, contact name, and whether it was published or not. But in addition to that, I can ask IMG to send me to the JGI genome portal and generate so-called download bundle. So I am clicking on the upload and export and save tab. And here I can find this download genomes button. If I click on it, it will send me to the JGI genome portal, which actually is the place at the JGI to download large amounts of data, not IMG. So if I click on this link, it will actually show me this page. I just click on it previously so that I can get the data. It's like a cooking show. I have a pan under the counter, and that's what it is. So it tells me that I selected 43 genomes to download via JGI portal, and that I won't get this data immediately. Instead, I will be contacted, I will receive an email from this email address. And if you have some very strict spam filtering options, then you may not receive this email. So it will be sent once the bundle, the download bundle, is generated and is available for download. And it also tells me that I can see, I can check the status of my download bundle using this link. So in addition to that, it gives you a warning that not all of IMG data is available for download from the JGI genome portal. These can be downloaded from GenBank using the GenBank accession or NCBI taxon ID or some other information. It gives you an example and it tells you how it will be expressed in the download bundle when you receive it. So it will tell you that for certain taxon IDs, again, these are unique identifiers assigned to each and every IMG data set by IMG, that for certain taxon IDs, the download is not available. So in this case, I waited long enough and I received the email from the JGI genome portal. And I clicked on the link that it provided. And here is the example of the page that I've got. So it tells me that the download request is completed, but I have this download file available, and this file is relatively small. It is half of a gigabase. But some of the files, download files, if you requested hundreds of metagenomes with all of the data, they can be many tens, if not hundreds of gigabytes. For these files, you can use Globus, basically the FTP download via Globus and other tools in order to get the file to your computer system. It also tells me that the data will be stored only for 2 weeks. After that, it will be deleted, and you will have to restart the download again. So it also tells me that not all of the genomes in my list were selected for download. This particular genome, the taxonoid, I will have to go to its genome details page, and from there to GenBank and download its GenBank record. We understand that it is probably not it's not like one-stop shop and so on and so on, but that's how it works. So if you are completely lost with all of this that we were discussing about, there is this option of help, right? So the help menu. Let's click on it and see what's there. So in this help section, there is a whole section about data usage policies, downloads, guidelines, and so on. Here, there is the section called download. It has data usage policy, data management policy, and also this downloading IMG sequence and annotation data. If we click on this, it will open a 12-page PDF file, which tells you in great details with screenshots about how you download the data, one data set, multiple data sets. It also tells you where to go if you don't see the files in the portal. It also tells you what to do with the downloaded files. Because if you have seen this, right, so basically it says tar file. What is it? Well, it is so-called tarball, and most Linux users know what it is and know what to do with it. But by default, Windows users don't have any installed packages that allow them to open these kind of files. And this tells you that this is what you need to download, this is the tool, this is where you can find it, and then you can open this archive, and this is the standard content of this archive. And that sort of ends the section about batch download of IMG data. And Rekha, do we have any questions immediately that we have to answer? There are too many of them, and we will have to write a whole new manual.
I guess it's some follow-up question because they might have missed it. What is the reason that some genomes are not available?
So the reason for, okay, so there are two main reasons. The first reason is that all the genomes in IMG, they were downloaded from NCBI and never annotated. So the IMG data is essentially the same that you would see in the GenBank record. So that's why we do not provide, because basically you can go to GenBank and download the same data. And we have limited bandwidth and we have limited storage resources. So that's why we are sending you to GenBank, because IMG data is virtually identical to what you see in GenBank. So that's one reason. The second reason is actually much funnier, and it applies to these private submissions of non-JGI data that were later made publicly available. So some users, basically, they opted out of posting the data for downloads. And that is the second reason why you may not have the genomes that's the second reason why we may not have the data in the portal for download. Anything else?
Can she show us again how she eliminated the 43 non-public genomes from the cart to get 26?
So basically, okay. You know what? that will bring the webinar to 2 hours. So how about this? I will write a detailed document about and examples of the expressions of the regular expressions that can be used to filter the data. And then we will post the document again, in this case, the download tips and tricks. And this will be sent to you together with the transcripts and recording and everything else. How about that?
Perfect, Natalia. Thank you.
Okay. So, okay, so this still ended the first part of the webinar about how to do batch downloads of the data and how to figure out which data is free for use and which one have restrictions on them, okay? Now, let's say that the last thing I'm interested in is this giant file, the tarball, because all I'm looking for is a few genes. For instance, I'm only interested in 16S RNA. Or I am only interested in protein coding genes with a certain function. Why do I have to do this batch download each and every time and then try to figure out how to get to these genes? Well, IMG is actually providing you with an export option in this case. You can find these particular genes and then you can export their metadata or their sequences without bothering with these batch downloads, okay? So IMG has multiple options of how you can find the genes of interest. So one of them will be presented in the first webinar of the second series, and that would be BLAST and other similarity searches in IMG. So again, we will post a notification, we will post an announcement. If you are interested in this option, please sign up. So, but it is not the only option for gene search in IMG. We are not sure whether how many people are interested in other options. If you are interested, please respond to the survey and say that I am interested in advanced gene search options in IMG, okay? So now in order to show how to export genes, a small set of genes and their sequences and their metadata from IMG, let's go to this genome of Bradyrhizobim elkanei, WSM 2783. If I click on it again, I will get to the genome details page of this particular genome. So this genome details page has a lot of different information, but what I am interested in this case is these general statistics. Remember the second category of the metadata in IMG, and this is the cumulative statistics. The total length of the genome assembly, almost 10 megabases. The GC content, 62%, and so on and so on. So the total number of protein coding genes in this case is almost 10,000. Let's say that I'm only interested in the genes for which we have a functional prediction. And this is only 6,800 of them. So all of this, as you can see, they're linked to something. Actually, if you click on any of these counts, you will get a list of genes in this particular category. So 6872 will bring you the list of genes with functional prediction, while 2679 will give you the list of genes without functional prediction. Let's click on this 6872. And while it is still loading, let's pay attention to what it says on the top of the page. On the top of the page, it says something completely unexpected. It says results limited to 1,000 genes. Why would that be? I want the entire list of 6782. So this is because by default, IMG is quite stingy with its data. It tries to limit you to the number of genes, to the number of homologs, to the number of contacts, and so on and so on. But you can change it by following this link, going to the preferences, and then changing these limits, increasing them. So as you can see, these are my IMG preferences right now. So it limits the number of scaffold result lists, number of homologs lists, and everything else. So it also lets you select the homolog minimum percent identity and so on and so on. So let's say that I don't want to see 1,000 genes. I want to see at least 10,000 genes on my list. So I change this the list results count from 1,000 to 10,000, and then I have to click on Save Preferences. And after that, I won't have to do it every time I log into IMG, because IMG will remember my preferences, and next time it will display the entire list of genes for me. Now let's go back to the genome details page and try again to get the list of the genes with functional prediction. So let's click again on this count of 6872 and see what happens. As you can see, these preference settings, they took place, and now I see the entire gene list. I can select all of these genes and add them to GeneCart and then do some filtering and so on. Or I can do some filtering right here. In this case, I will just use plain text filtering on the gene product name, which is the name of the protein, the predicted function of the protein. So I will select this column gene product name. I will keep the filtering option as text, and I will type here cytochrome. So this will look for, basically this will select out of this list of 6,800 genes, this will select only those that have cytochrome, the word cytochrome in their annotation. So if I click on apply, I only have the genes that have cytochrome in their annotation now. So I can click on select all. And then I can click on add selected to GeneCart. Again, carts in IMG as workspaces, these are the places where you store either permanently or temporarily the lists of objects of interest, such as the genomes that you intend to work with or the genes that you intend to work with. So in this case, for instance, I was looking for cytochromes. I could be looking for all genes that have that are annotated as aminotransferases or aminoacyl tRNA synthetases or something else. So now I have GeneCart, and I have 146 genes in my GeneCart. As you can see, this is the default table, and it shows you the gene ID, the locus tag, and the gene product name, essentially the same information that you have seen in the list. It also tells me that all of these genes in my GeneCart are coming from the same genome with this ID, with this genome ID and with this name. Now, let's say that I'm not interested in all cytochromes. Let's say that I only want the cytochromes that are integral membrane proteins, which means that they have transmembrane helices and probably at least 2 of them. Now, can I do this in IMG? Can I find these genes in IMG? Well, and the answer is absolutely. To do this, again, you would use the table configuration options. The same way in GenomeCart, you would have table configuration options. There are table configurations options in the GeneCart as well. They're on the bottom of the table. If you scroll down, here it is. So again, there are several categories of the fields that you can display in this table, and they are divided into gene field, scaffold field, function field, and project metadata. Let's focus on the gene field. As you can see, I can add the start coordinate, end coordinate, the strand of the gene, the amino acid sequence length. This is something I may be interested in. I may also want to see whether this gene have any and how many transmembrane helices. So I will select this field as well. You may be also interested in, for instance, the scaffold length, scaffold GC percent, or you may look at whether these genes are assigned to any of these protein families. For instance, let me select the PFAM in this case. So after you selected the columns that you want to see in the table, again, you have to click on this display genes again. Otherwise, IMG won't know that you want to see additional data in the table. As you can see now, the table has been updated, and now it has this column amino acid sequence length, transmembrane helices, and also PFAM annotation. if you remember, I wanted to see only the proteins that have at least 2 transmembrane helices, indicating to me that these may be integral membrane proteins. How can I do that? So first of all, let's deselect all of the proteins in this table by clicking on clear all. The second step is again to filter the table. In this case, I will be filtering it by selecting the column transmembrane helices, again going with the regular expression, but in this case, it is very simple numerical filtering. I would say that I want only the genes that have at least 2 transmembrane helices. So I will say greater or equals 2. So then I can click on apply. And now my table is filtered, and all those proteins that don't have any transmembrane helices or have less than 2, they were basically they were hidden. So let's say that I want to export the content of this table, and again, so in this case, I am only using these columns, but you could have included all of the data that you see in this table configuration on the bottom. So let's say that I want to export the genes that have more than 2 or more transmembrane helices. I will click on select all. And then again, I will click on the export button. Again, it will tell me that there is this file that can be opened with Excel. And now I will have to stop share and switch to the sharing of another window to show you the Excel file and its content. Can you see the Excel file, Rekha?
Yes, I can.
Okay, great. So as you can see, again, this is the export of all of the content of the selected genes from my gene card. And it has the same columns that you had in gene card. So it has the gene ID, the product name, the genome, the amino acid sequence length, the number of transmembrane helices, and it also has this PFAM annotation, where you have the PFAM ID as well as the name of the, basically the name of the PFAM that we are looking at. So again, it is tab delimited file. So basically it can be parsed, it can be in Unix, it can be parsed with in many different ways. Again, the most important column in this file is here. Just like every
Natalia,
IMG.
I think
Yes?
you're sharing the wrong screen. The wrong Excel
Christ.
file.
Okay, thank you. Let me try again. It's the Zoom thingy that I have to. Oh. Okay, I have too many Excel files open, sorry. Can you see now the Excel file for the genes?
Yes.
Okay, so again, so to repeat myself, so basically the gene product name, the amino acid sequence length, the number of transmembrane helices, the PFAM file, the PFAM annotation. The most important column again is this gene ID column, which is every gene in IMG, like every object in IMG, has a unique numerical identifier. That's how you can find it the easiest way, and that's how you can load the content of basically these genes back into gene card if you want them restored there. Okay, so now let's go back to IMG and let's hope that I don't screw it up. Can you see the browser again? Hello?
Yes.
Yeah.
Okay, thank you. So that's the export of the metadata, right? So basically, remember, this was the export of the metadata. In addition to exporting the metadata, I can also get the sequences of this particular set of proteins, not the entire genome, but only this set that is in my gene card right now. And not just the entire set, but only those ones that are selected. To do that, I will click on the tab Upload, Export, and Save. And here you have, we have multiple options. I can export a FASTA amino acid format. This will be the amino acid sequence of the selected genes. The second option is the nucleotide sequence of the selected genes. And here I can elect to output additional nucleotides either in the upstream region or the downstream region. Let's say that I want to see the 300 nucleotides upstream of the gene. So then you click on the, so you need to read the warnings here, right? That if you have a lot of genes and you want all of them exported, it will be slow, which means that if you have more than 100 genes that you want exported, you will get an email. And this option will be also available only if you are in MER. So let's say that I want the nucleotide sequences of the genes plus the 300 base pairs upstream. So I will click on this show in export format. As you can see, what on the page, I have this multi-FASTA format file. I have the nucleotide sequence of the gene itself, where the stop and start codons are highlighted in red. The sequence itself is in black, and the start and the stop codons are highlighted in red. And then in green, I have this additional 300 nucleotides that I wanted of the upstream sequence. The same way I can download, I can export the sequence of the protein, the amino acid sequences of the protein coding genes. And the same type of export capabilities for a small set of selected objects is available for the scaffold and Contig card, basically for the scaffold card for the export. And that ends the second part of the demo. Are there any questions right now?
Not pertinent ones.
Okay, then I will say thank you to all of the participants. Again, a reminder that this webinar is recorded, that you will get a recording, the link to the recording, and the link to the chat with the answers to your questions. We will try to come up with a document describing basically, again, the tips and tricks of what kind of regular expressions you can use to filter the data. Again, there will be a link to this survey. Please respond, because otherwise we won't know which topics are of particular interest to the webinar participants. Thank you so much for participating.